Okay, hello everybody. It's John Johnson from NSF. I'm Executive Director of our UK headquarters in Kirby Moorside, and I've prepared for you today a relatively short summary of the proposed changes to EudraLex Volume 4, Annex 1, to try and give you an early insight into some of the major changes within this part of the GMPs, and also some interpretation on how that may be inspected or enforced by the regulatory bodies. So we're going to spend about 30 minutes or so making sure we all understand the reasons for the update, the key parts of the update and the process for making this new annex approvable. And we're going to talk about how to react to those changes, how those changes may affect you in your organisations, and how you may best and most proactively ensure perpetual GMP compliance against these expectations. So a very warm welcome to everybody from France, Denmark, Netherlands, UK, UAE, Sweden, Norway, Italy, Belgium and the USA. Phenomenal amount of interest in this subject, such a high degree of interest that we're going to be running this webinar twice, which is actually a record for NSF. So some fantastic attendance and obviously a lot of interest. I'm going to be moving through the slides fairly quickly so that we can concentrate on the key messages. So I'm not going to say too much about our organisation. I think most of you know us very well. We're specialists in education programs, auditing, certification, vendor assurance, and also consultancy across GMP, particularly in transformational projects that involve culture, remediation against quality systems, or assisting in new facility builds or improving business performance. So I'm not going to say too much more than that. Looking through the attendance list today, many of you have worked with me or know me, so I'm not going to say too many words about this. Just suffice to say that I'm the NSF course leader for the A to Z of sterile manufacturing course that we run as a public course in Amsterdam and Manchester 3 times a year, and also the same course internally at clients' premises several times throughout the year. So we've got a deep understanding within our organisation of the GMPs, specifically in this case in steriles GMPs, and we have some fantastic team members who have had experience either as qualified persons, quality directors, technical directors, site leaders, or indeed from within the regulatory agency. So what I'm going to be talking about over the next half an hour or so is why is this particular annex being updated? Why is it being updated now? What do we hope the updates will achieve? What are the current areas that seem to be under significant discussion that's causing some delays to the final approval of this annex? And of course, what happens next and how might you best respond to it? Well, the first thing to mention, we're now on slide 5, the first thing to mention is that ultimately sterility assurance and the quality assurance of sterile products in general depends upon good science, good compliance, effective leadership, and a deep commitment to ongoing education. And a combination of science, compliance, leadership, and education is important to reflect in the regulations, and of course is going to be important to reflect in your site quality management systems, your policies, procedures and records that confirm that you're meeting these expectations. So the first reason why this is being updated is actually around the science. It's actually been nearly 20 years since Annex 1 was first written, so it's really due an update. You know, there's been a number of changes in those 20 years, a number of non-mainstream technologies that have emerged, the growth of advanced therapy medicinal products, the growth of radioisotoping of parenteral formulations, number of advances in form-field seal, and of course there's been a number of advances in the way that we design, validate and monitor our sterile facilities. So the first reason, of course, was to make sure Annex 1 reflects those new technologies. The second reason for updating it is perhaps more complex. There's very much been a trend over the last 20 years to set up new manufacturing locations as centres of excellence. So that's meant that our global supply chains have become much, much more fragmented, much, much more global, and in some cases the transfer of manufacturing locations to new locations around the world has led to a discontinuation of the inherent knowledge, know-how and know-why around your particular production process. So there's a perception from the regulators and from within the industry as well that this trend of global supply chains, of fragmenting the supply chains to new locations, can sometimes lead to discontinuous investment in the knowledge of how to make sterile products. How do we know this to be the case? Well, speaking to the Annex 1 rapporteur, Andy Hopkin, and speaking to a number of other agency representatives within Europe, there is a real concern at the GMP deficiencies that are being found in steriles manufacturing companies around the world. That many of those GMP deficiencies are relatively straightforward. They're not technicalities, they're not particularly complex to fix, but there does seem to be a theme that some of the very basic requirements of making a sterile product are not in place, not embedded, not hardwired into the quality system, and in many cases do not have effective proficiency tests or effectiveness checks to confirm that those GMPs are actually working. So there was a feeling that Annex 1 should actually have some more detail, better clarification to actually assist those new locations so that they understand perhaps in greater clarity what the expectations are within the EudraLex Volume 4. The other key change for us as well, of course, two absolutely crucial CGMP requirements, ICHQ9, Quality Risk Management, and ICHQ10, Pharmaceutical Quality Systems, have been around now for 15 to 20 years. And there was a perception from the regulators that we needed to align Annex 1 with the requirements of ICHQ9 and Q10. What does that really mean to you in practice? Well, it's going to mean that you're going to need to take both a proactive and reactive approach to risks within your sterile process, making sure those risks are properly identified, assessed, evaluated, ranked, root causes well understood, and the correct CAPAs and CAPA effectiveness checks are performed. So the new Annex 1 is going to make some very strong links between steriles manufacturing and the requirements of ICHQ9 and Q10 in particular. There's also a perception within the industry and also within the regulatory bodies that there appears to be in the sort of 20-teens, the early and middle part of the 20-teens, that there is a high turnover of expertise. So a number of people that have been very experienced, very influential, have been mentors to critical position holders within their organisation, appear to be leaving the industry, and there is a concern that we don't, as an industry, have an effective successor management plan that involves mentoring, practical and also theoretical education in this absolutely critical area. And there's also a perception as well that some of our sterile products, in fact many of our sterile products, are now viewed as commodities. The margins are very tight, and that means that sometimes we're driven to take certain risks or be blind to certain risks that could be seen as unacceptable by a third party. So there has been an awful lot of change within that 20 years, both in terms of the expertise within the industry and also the commercial imperatives. Equally, I think everybody that I've spoken to within the agencies and as quality directors and leaders within our organisation, there is a feeling that we really don't always do a good job in making sure that our education programs are linked to the particular risks in our process or in our organisation. That sometimes education lacks detail, staff don't get the time to practice and be sure that they're comfortable with the processes, so that ultimately we can put in place proficiency tests, both on induction and also periodically, that will assure, for example, aseptic behaviours, the quality of aseptic connections, the quality of aseptic interventions and sampling, and a variety of other critical tasks. And during inspections, with work that we do and also within the regulatory bodies, we're often kind of surprised that people have the know-how of what is expected. They know their procedures, they know their policies, but when asked about alternatives, or when asked about change or how change may affect the degree of sterility assurance, we are often surprised that the know why is not there. So Annex 1 is really hoping to kick off a fairly standard definition of how to make sterile products, but also develop an approach that reinvests in the human capital and the knowledge within this particular sphere of pharma. So moving on to slide 6, sterility, of course, is not an absolute. We all know that this is based on a probability that depends on all parts of your quality management system. And specifically, the good design, effective control, and proper, thorough, comprehensive, and ongoing monitoring of the sterile core and the sterile processes. And we know, of course, that we can't depend on luck or good fortune. We have to make sure each of those small details amongst design, controls, and monitoring combine to give you the sterility assurance that you're expecting and can demonstrate. So what are these updates aiming to achieve? First of all, of course, the new Annex 1 has got to reflect a number of new technologies. So there's going to be a number of references here to and more detail to things like form-fill seal or blow-fill seal technologies, a little bit more information about ATMPs, also small batch sizes, specials, formulations, and perhaps more detail as well on the way sterility assurance is assured in non-mainstream technologies. There are also, of course, as I'm sure you realise, some parts of Annex 1 that contradict each other, some where the definitions are somewhat opaque, and there's been an awful lot of work done to try and clarify those definitions, clarify the expectations, and make it easier to read and actually put in place and enforce. But if there was one key message I wanted to get over to you really in this very short webinar, was that the Annex 1 is going to drive us all to think very hard about the scientific rationale that we've applied to the design of our facilities, the control of those facilities, and the way we monitor them on a day-by-day, month-by-month basis. Because the industry is seeing that there are a number of different ways of achieving the same thing, but they're not seeing the industry's justification for doing it that way. They want to see how you've assessed the risks, and one of the key documents that's likely to be much more frequently requested is your quality risk assessment for sterility assurance of your key formulations, actually showing a line-by-line process definition, a line-by-line process flow chart that shows where the product is open, where it's closed, where there's certain filtration or sterilization steps, and obviously identifying where the key risks are. And ultimately, by doing that, identifying, assessing and mitigating risk, everybody is looking to improve the quality of sterile products from the industry. So Annex 1 is being worked on just now with a variety of people led by Andy Hopkins from the MHRA. He's working with the EMA PICS working party. It was the best part of 2.5 years ago now when the first draft concept paper was issued by MHRA, and about 6 or 7 months ago the final, or at least the next full draft, was issued to the inspection working group. And ultimately we're expecting that concept paper to be published sometime in the next quarter. Really, our expectation was the first quarter of 2017, but there are some areas that are still being discussed that could lead to a small delay against that. But our current understanding is that the first concept paper that's open to review by the industry will be available within this next quarter, first quarter 2017. A key quote that has come from the MHRA symposium and often repeated within the industry was from Andy Hopkins, and he said that he expected no adverse impact on industry with respect to either your resources or the costs. And that's a great quote and one to hold on to and take some comfort from. But of course, it really depends upon the level of GMP compliance that you're operating to at your facility at present. So I would certainly recommend, when the concept paper is published, you do a line-by-line review and risk assessment against your quality system and your sterile processes to ensure you're meeting those requirements. Obviously, if you're not, there could be an impact on your resources or costs. I would certainly recommend that to you. Moving on to slide number 9, I did a fishbone diagram for our recent journal number 38. So the NSF journal came out about 3 weeks ago now. And what I tried to show on this fishbone is the 12 sections of Annex 1, and to give some basic information about the key modifications to Annex 1. And I know many of you already received the journal. I think that we're going to make sure that these slides are available to you as part of the webinar. And I'd recommend that you just go through these. I mean, most of them, as Andy Hopkins said, are just clarifications. But there are 2 areas in particular that still have some controversy. So I'm going to go through those in a little bit more detail. So if we looked across the 12 sections on slide 9 and we separated out the 6 key messages, what would they be? So the first one I think I've mentioned already, there is going to be a hardwired link to ICHQ9, to your quality risk management process. And what I would really recommend here is 2 things. That you look very, very hard at the way you do proactive risk management, the way you use risk management to define processes, validate processes, and decide on the rationale for monitoring and sampling processes. Making sure that the areas of high risk are well understood and mitigated wherever possible. And making sure that you use some form of risk register that uses the ranking tools within ICHQ9, backed up with scientifically justifiable technical reports that explain your rationale and your decisions are going to be really important. Of course, the second part of risk management is the reactive one. So when things go wrong, when you have an unpredicted issue, an adverse event or a customer complaint or a GMP deviation or an out of specification, out of trend result, how do you respond to it? How do you make sure you've fully understood the most likely root causes, that you fully understood where the problem started and ended, you fully understand the product impact assessment, so that ultimately by effective root causing you can put in place CAPA that prevents recurrence of that particular issue. So generally people are seeing that in many cases, for example, the GMP deviation reports surrounding issues in sterile processing could be significantly better if ICHQ9 was implemented in full. That also takes into account really key message number 2, making sure that you utilize your critical position holders, your subject matter experts within your organization to be sure that you've come to the right product impact assessment with the right root causing. The third key message I would say is that Annex 1 is really going to stress the importance of education rather than training. We often say we train our dog, but we educate our children. And education is much, much more than just a training program. It's really about customizing the messages, making sure the messages are understood, the know-how and the know-why, that people get the chance to practice, they get the chance to work alongside experienced people, and those experienced people get to actually verify the proficiency of those new staff before they're left on their own to continue. Because we all know, particularly in steriles manufacturing, the attitude, behaviors, the demonstration of decision-making ability amongst sterile operators and sterile manufacturing staff and QA staff is absolutely critical. The people aspect cannot be overstated for this particular dosage form. Interestingly enough as well, the Annex 1 is going to also give some pointers on how attitudes, habits, staff behaviors should be enshrined within education programs, should be enshrined in the way leaders lead and educators educate, making sure that people make the right decisions when they're not being watched or when they're not being supervised or when they're working out of hours. Because ultimately some of the key risks can occur in some of the simplest situations, particularly on the shop floor during open or semi-manual processes. Key message number 5 is all around the separation of staff from open or critical stages in the process. There's going to be a lot more information here about effective facility design and how to properly separate the key risk, which is of course humans, from open processes or vulnerable stages in the process. And then of course the 6th message, as I mentioned, is going to cover some detail on the non-mainstream processes. So what are the 2 areas that are actually holding up the approval of the concept paper and issue of the concept paper? It's really surrounding these 2 issues, and the first one is commonly called PUPSIT, a very interesting acronym for this, PUPSIT, P-U-P-S-I-T, which means pre-use post-sterilization integrity testing. So there is some discussion at the moment within the working party as to whether pre-use post-sterilization integrity testing is going to be a requirement. There's a lot of discussion about the value of the supplier's certification on integrity testing, the ability of the filters themselves, and we're particularly talking about the terminal 0.2 micron sterile filters here, the ability of those filters to withstand damage during sterilization, and also the integrity test methodology having a risk of either damaging or contaminating filters. So ultimately we want to be able to come up with a requirement that is practical, takes into account the technologies at the filter manufacturers, the certification, and also the integrity test methods that are employed. Because ultimately, of course, we want to be handling or subjecting those filters to manipulations and tests as little as possible, providing we can demonstrate effective integrity of that filter medium and the filter housing. So there's still a lot of discussion about that, and watch this space. We're going to be updating you on the progress of this in our A to Z of Sterols training course. I'm almost certainly going to be doing another webinar in the summer on this as more detail becomes available. And there's also going to be information shared on our LinkedIn pages. And also we're going to be launching an NSF app at some stage this summer. So there's a number of ways you can access the final decision on this one. The second one has been an issue that I think has troubled the industry for some years now. I mean, considering all of the different types of container closure designs, whether it's glass or plastic or vials or ampoules or syringes and such like, there is a lot of discussion about the effectiveness of the technology to provide a 100% container closure integrity test, or whether there should be some offline sample test for certain types of containers. So there's a lot of discussion going on at the moment about whether the technology allows us to do this and what those new developments might do to our regulations and our perception of risk. Interestingly enough, I was at PDA Europe, when was it? It was last summer, I think it was last July or August, and one of the speakers described how in the next 5 years there should be technology available to be able to do a 100% sterility test of every unit that is non-damaging or non-influential on the unit. Now, can you imagine what a difference that technology is going to make to our way of making sterile products? In that, online, you will be able to verify the sterility of each and every unit. That's the type of technology that is coming down the pipeline, so we need to be aware of that too. So ultimately, if you look at the Sterols facility, how are you going to prevent issues? How are you going to maintain compliance within your sterile core? I say this a lot, and there's no apologies for saying this. There's nothing better than actually walking the job. You're going to see this a lot more with the inspectors. They're going to spend more time walking, witnessing, reviewing batch records in situ, talking to operators, looking through the window of your aseptic core, because they realize that by walking the job, doing the Gemba, going to where the action is, it can be a lot more insightful in the level of compliance compared to just reviewing documents. So if they're going to do it, guys, I think maybe we ought to be doing it as well. Of course, as a practitioner within the sterile core, it's in your interest to have a deep understanding of everyone's jobs, have a deep understanding of what's troubling them, what's hard for them, what's complex, where the likely errors are going to occur. And of course, by asking those questions, you're showing that you care. You develop a relationship where people will talk to you and tell you where the issues are, and you can start looking at those issues and applying ICHQ9 quality risk management to those issues so that you're actively seeking, evaluating and mitigating those risks in full. Of course, again, underpinned by Annex 1, the new Annex 1, it's going to be very important to structure and be very thorough at looking at the key root causes to problems. Use the tools that are available in ICHQ9, utilize human error tools, understand in full why a human error may have occurred. You know, our research says that there's typically between 3 and 9 contributory reasons to every human error. If you're not fixing those contributory reasons, you know it's just going to happen again. So be obsessive. Know the job, know the people around you, develop that trust, and make sure that you're continually verifying and monitoring your quality assurance risks. So what would be the next steps? Ultimately, please keep in touch with us. Don't let the hand sleep. You know, be good at each of these 6-fingered issues that I've put on the top right of slide 13. be very active in putting these things in place in your company. And when Annex 1 concept paper comes out, I would really recommend you perform a line-by-line gap analysis for your operation. Look at the 4 areas that impact sterility assurance, as we talked about before, leadership, science, compliance and education. Use those ICHQ9 tools. Understand the root causes of the findings that you identify. Put in place CAPA. Consider how staff training could prevent a reoccurrence. And obviously look for some form of effectiveness protocol to be sure those changes are implemented. Of course, we can help in these areas, particularly in education programs, in doing gap analysis and GMP audits. And of course, whenever you make a change, think hard about how that might affect culture, how you fit behaviours to your new expected process, how you motivate or make people able, or remind people, or make people habitually follow these new changes, so that a transformational change is actually embedded and actually reduces risks rather than adds to it. So we can help in that if needed. We're also very active again this year in our public courses. These are the key courses that we offer in this field, both in Manchester, Amsterdam and in York. And of course we bring all of these courses in-house, customized to your technologies, your requirements and your delegates as needed as well. So hopefully you can take advantage of those. We also have a very active webinar program again this year, with the next 2 actually being very complementary to Annex 1 and Sterols Manufacture, with Martin's webinar on March the 3rd covering GMP behaviours, how to break old behaviours, bad habits and make the new ones stick. And then my webinar on March the 27th about making sure you prevent Groundhog Day, you prevent repeat issues. So hopefully they'll be of interest to you too, and of course they're all free. So anyway, thanks for listening. I think I've taken about 33 minutes for this webinar, so 10% extra this time round. I hope that was really useful to you, and I'll make sure that you're kept informed of the changes to Annex 1 as they happen. Please feel free as well to go to our website and look at my videos and webinars that could be extremely helpful to you in your day-to-day work back at base. So hopefully that was of interest to you, and that technology worked well and everything was clear. Please feel free to email me anytime. I'm always here to help you, and I love to talk about this subject, as you know. Enjoy the rest of your day, and I'm hoping that was good for you. Take care, everybody, and best of luck.